Good afternoon. Welcome back to KAIST Emerging Materials Symposium 2020. So in the afternoon session, we have three great speakers, and my name is Yeonsik Jung. It's my great pleasure to chair the afternoon session. So first speaker is Professor Bin Liu. I will briefly introduce her. Professor Liu is the professor and head of the Department of Chemical and Biomolecular Engineering at the NUS Singapore. She received a BS degree from Nanjing University and a PhD degree from NUS before her postdoctoral training at the University of California, Santa Barbara. Professor Liu has received many highly prestigious awards, including Singapore President's Young Scientist Award, L'Oreal Women in Science National Fellowship, Asia Rising Star, and also she was named one of the top 1% highly cited researchers. And currently, she's serving as the deputy editor for ACS Materials Letters. Okay. So please join me. Welcome, Professor Liu.
Okay, thank you, Chairman. Before I start, I would also like to thank Professor Kim for organizing such a wonderful event. I would like to take this opportunity to share with you a unique type of luminescent materials with aggregation-induced emission. And today I will primarily focus on their biomedical applications. Luminescent materials are actually everywhere in our daily life, from the optoelectronic devices to the biomedical research, security, and solid lighting. They really make our life very colorful and exciting. But as a material scientist, when we zoom into each of the individual materials, we might find something striking. So if I take this DPT molecule as an example, this molecule is very well dissolved in organic solvent to be highly emissive. But as far as we're able to induce the aggregation formation for these molecules, for example, we add water into the THF solution, what we observe actually is a gradual decrease in the fluorescence and eventually reaching a complete dark state. And if we look at the molecular interaction, it's very clear that there is a strong pi-pi interaction among these molecules, so that it is really one of the main reasons to cause a fluorescent quenching, and which is not practically useful in many applications, such as in light-emitting devices or for biomedical applications where molecules actually naturally aggregates in the aqueous media. So what we are interested in is a new type of molecules with aggregation-induced emission, and TPE is one of the typical AIE molecule. And this molecule actually has the key feature of molecular rotors. So in the molecular state, this molecular motion actually consumes most of the excited energy, and therefore these molecules are not emissive. But once these molecules are brought into the aggregated state, being in the nanoparticles or fabricate into the films, this restriction of molecular motions actually will boost up the fluorescence, and because of the rotor structures, these molecules are not able to come into very close proximity to quench the fluorescence. And that's why we're able to produce molecules that are highly emissive in the aggregated state. Let me give you one specific example in terms of application. Whenever we open any catalog, and here actually I took the organelle stand dyes as an example. So these dyes are often sold as the DMSO stock solution. They're highly emissive. But once we add in water or we dilute these solutions with cell culture media, most of us didn't notice that actually the fluorescence is almost quenched before we use them actually to stain the cell and watch the fluorescence. That is a clear indication that in reality, we didn't really fully utilize the great potential of luminescent molecules really to serve the best purpose as they can. Now, if we look at AIE molecules, they behave exactly opposite to the traditional fluorophores. AIE molecules are not emissive as molecular species, but upon addition of water to induce the aggregation formation, they become highly luminescent, and the same phenomenon is also observed in the solid state. So that really makes them very special if we look into their applications in the light emitting devices or simply for biomedical applications. Now, on this slide, I show you some of these molecules. They're highly representative. Actually, there are many of these molecules available in the literature. But what I want to show here is that how simple design of this molecules actually can form a family. They in very polar solvents, and we can easily actually fine-tune the molecular structure to achieve the light emission from UVBs to near infrared, and now it's even into the much longer wavelengths. And all these molecules can be induced to be highly emissive in the aggregated state. So over the past 10 years, we're pretty much fascinated about these two extreme phenomenon, that AI molecules are not emissive as molecular species, so that they can be used to develop light-up probes that do not have any background signal, which will allow continuous monitoring a lot of biological processes that are previously not possible. And at the same time, if we look at the aggregated state, these are the molecules which show great potential to be designed into nanoparticles that are highly emissive, which allow us actually to track a lot of biological processes and to conduct medical imaging, and then further we upgrade them into the therapeutic nanomaterials. So today I would actually primarily focus on the aggregates, but I will start with a little bit of brief introduction of the AI light-up probes. So we designed two types of AI light-up probes, fully to really utilize the great potential of AI molecules as molecular species. They're not emissive. So the first type actually is based on the specific lock and key interaction, where we decorate the AI molecules with specific recognition elements, which upon interaction with the analytes, because of the restriction of the intermolecular motion, we're able to actually turn on the fluorescence as the indication of the presence of the analytes. And the second type is based on very specific reactions, for example, enzymatic reactions, that actually allows the turn on of the molecular probe in response to either enzymatic reactions or chemical reactions or any other reactions that are highly specific in the biological processes. So with these two platforms, able to actually produce a series of molecular probes that allows us to detect, for example, the proteins on the cancer cells and molecular species that exist in the biological system, and the reactivity of the enzymes and how this enzyme reactivity is associated with the drug screening, etc. And gradually, we ask ourselves one question: would it be possible that if we design some special molecules which will not only offer the optical changes, but also at the same time incorporate with the therapeutic functions, so that we're able to upgrade our first generation molecular probe into the second generation, which is the theranostic probes that not only offer the sensing imaging functions, but also the therapeutic functions. So this motivates us to look into the molecular design. And one of the examples that I would like to share today is AI photosensitizers. So many of us actually are fully aware of how the photosensitizers work for a traditional molecule. Actually, as far as we're able to facilitate very effective intersystem crossing, the triplet state will react with oxygen to form the singlet oxygen, which is a radical that is harmful to tissue cells and bacteria, etc. Now, if we look at traditional photosensitizers such as porphyrin, many of them actually have planar structures, and therefore these molecules, when they are brought into nanoaggregates, they tend to have very low light emission as well as reduced photosensitization capability. So what we propose here is to take advantage of the AI molecules in the aggregated state when majority of these non-reactive decay pathways are blocked, so that we are able to create a scenario where we're able to simultaneously enhance both the fluorescence and the singlet oxygen production, as far as we're able to make sure that the energy difference between the singlet state and triplet state is reduced to minimum energy level as much as possible. So let me use a series of molecules to explain how the AIE photosynthesizers can be designed and make a difference in terms of performance. So we started with tetraphenylethylamine, which is the molecule that actually gives light emission in the aggregated state, blue color, with very weak or almost no capability in producing oxygen. So the first step we did was to add the donor acceptor structures and then add on the bridge with the purpose to separate the HOMO and LOMO energy levels of the molecules so that we're able to reduce the energy difference between the thinly straight and intersystem crossing. And we're able to actually observe the thin oxygen production for these molecules. And subsequently, we add in the pi bridge to enhance the light absorption. We also changed the acceptor, trying to shift the emission of the photosynthesizers to the long wavelengths. We also produced a new approach, which is the polymerization approach, that is able to increase the energy levels for both singlet and triplet stage so that the chance and probability for the intersystem crossing becomes much higher as compared to small molecules. Now, with all these designs, if we compare our AIE photosynthesizer with the state-of-the-art photosynthesizer available on the market, what we discovered was something really exceptional, that AIE photosynthesizers almost shows linear increase in brightness and the capability to producing the oxygen as far as we increase the number of molecules encapsulated in this nanoparticle, and which compares really favorable, much more favorable as compared to traditional molecules where they have a saturation followed by a decreasing in both intensity and the capability to produce the oxygen. Now, I want to share with you one of the interesting examples where the AI molecules actually can be used for sensing, imaging, and therapeutic functions. And this example is based on one of the issues that we have to address, which is the resistance of antibiotics. And we use antibiotics a little bit too much, and antibiotics were designed not to be really cellular permeable, and therefore, when bacteria actually entering the cells, that actually creates a scenario that's very difficult to kill those bacteria, and that will cause a lot of consequences in terms of how the disease will develop. So in this case, we take advantage of a special reaction, which is the metabolic labeling. That is actually a naturally occurring reaction in the living system. We just need to make sure that the molecules we feed into the bacteria actually contains a metabolic motif. In this case, is the amino acids. So that the bacteria, as they divide and grow, they will automatically incorporate this molecule onto their surface at the end of the side chain of their peptide glycan. So these molecules, when we design it, we took consideration of few factors. First, it has to be very simple, very small in size, so that the biological system will just like it as one of the nutrients that they will need to grow and to divide. And second, this molecule has to be not emissive so that we're able to realize a turn-on response in terms of capture specific bacteria. And third, we also want this molecule to be really cell permeable so that it can go inside of the cell to capture the bacteria and then kill it, and hopefully will not produce anything harmful to the cell itself. So how do we know that this molecule actually can be incorporated onto the cell surface? What we did was to actually take out the peptide glycan, and then we digest it to form the dimer, and we look into the molecular mass so that we're able to capture exactly the molecular weight as an indication that actually the reaction occurs in this way and with this kind of particular sequence of the peptides. And we did the staining of the bacteria, and we co-stain with the Holchester, which will actually stain the interior DNA of the bacteria so that we know exactly these molecules are able to actually label the surface of the bacteria in a way that's quite effective. Now, if we incubate our molecules with the bacteria directly, we will be able to observe that in about 15 minutes that the molecules will naturally react with the bacteria, actually incorporate into the bacteria, which reaches a saturation in about 30 minutes. Now, if we incubate the bacteria with the cell, so on this bright field image, you will see a big cell, and inside of the cell, you will observe a lot of red dots. Each of the red dots actually is a bacteria. So this molecule is able to enter the cell and then label the bacteria. And now, if we follow the fluorescence image and then shine a white light to activate the photosensitizer, we will be able to kill this bacteria inside of the cell. And detailed analysis reveals that if we use 20 micromole molecular probe, we're able to successfully kill the bacteria inside of the cell, and yet the host cells remain alive. And the main reason is that this photosensitization produces radicals, which has very short lifetime and also a very small radius of interaction. Because these radicals are surrounded or are really on the surface of the bacteria, that's why they're very effective in killing the bacteria, while they're actually floating in the cytoplasm of the cells, and therefore they're much less effective in killing the whole cell. So with this kind of design, we're able to use a simple molecule, go inside of the cell, and then capture the bacteria, let us see them, and then help us to kill them. Now, the same experiments when we conduct in the living system, basically we have poor mice that has been infected by the bacteria, and then we inject the molecules from the tail vein of the mice. This molecule is very smart in the way that it will circulate around the blood system and reach this bacteria site, will automatically conduct the reaction and eliminate the bacteria, which guide us actually to conduct the photodynamic therapy. And we did the organ analysis, and we also trying to identify the total number of the bacteria before and after treatment, and we also did the tissue slice, trying to identify and also prove that there's indeed a huge difference with and without the treatment. So through this kind of design, we're able to actually come up with molecules that are not complex, but yet very smart, that the molecule will do everything as we need to solve the problem that we really face on a daily basis. Okay, so now I switch the gear to the other side to focus more on the AI nanoparticles. AI nanoparticles actually is the first commercial products for the whole AI family and AI research. And AI nanoparticles, or the AI dots, actually refers to very small size organic nanoparticles with the AI molecules at the core and the polymer at the shell, which are very precisely designed and produced with surface functionality that allow us to study biological processes, tracking cells, and visualize the cell-cell interactions, and also serve the purpose for imaging-guided therapy and imaging-guided surgery. So we started to fabricate this nanoparticle using a self-assembly approach, which is quite straightforward, that organic molecules will be wrapped at the core because of the amphiphilic block polymers. And we can fine-tune the functional groups, in this case, the amine groups, or can be carbonic acid groups, can be any other groups of interest, onto the surface of nanoparticle. The whole process actually took about 30 seconds, which is a very fast self-assembly process. And we can further conjugate a lot of recognition elements to make our nanoparticles highly seductive. So this process actually is very simple, straightforward, and every student can produce. But the issue is that not every student will be able to produce exactly the same size and same shape every time they did the experiment. That will create a problem for subsequent biomedical research. So driving by that, we move on to develop an automated system. It's really trying to eliminate the human error and trying to make our experiments as reproducible as possible. So we use a mini fluidic system where we have two pumps that carries the aqueous phase, and a central pump that is the organic phase, which is which contains the polymeric shell as well as the molecule that we wanted to wrap into the nanoparticle. So by controlling the flow time, by controlling the flow rate, we will be able to control the flow time. And very luckily that if we're able to manage the mixing time among the two streams is in the range of about 5 milliseconds, we're able to produce nanoparticles with 20 nanometer And when it comes to 10 milliseconds, it will be 40 nanometer. When it comes to 15 milliseconds, about 90 nanometer. So this really allows us to produce nanoparticles with high precision, high reproducibility, and more importantly, really in a large batch, so that can supply to our subsequent biological experiments. And we further actually spent quite a lot of time to build the phase diagram. We're really trying to precisely understand and determine under which conditions that we will make the best particles as the best as we can. And interestingly, there's only a very tiny zoom in this phase diagram that will allow us to get high quality nanoparticles. And beyond the zoom, either the particle will be too big or out of the shape, or you simply form a homogeneous solution that will not get any nanoparticle. Now, so after we optimize this system, we conduct multiple batches of experiments, and we ask many students to try out. We're really being able to produce organic nanoparticles with very narrow size distribution and also almost constant size at each of the trial. And we're very happy that with this millifluid system, we're able to produce nanoparticles that are very stable. They can be stable for weeks, and now we find out actually they're stable for up to a year, which that really meets the standard of the commercial applications. And what struck us most was that when we conduct analysis for each of the nanoparticles by collecting the photon image in 100 seconds, and we compare the organic nanoparticles versus the commercial quantum dots. And this is based on the similar size of the nanoparticles, which include both of the core as well as the shell. And we find out for organic nanoparticles, the total photon emitted per unit time actually can be as much as high as the 10 to 40-fold more than the commercial quantum dots. So that really triggered us to spend a lot more time and energy on organic nanoparticles, because they can be designed to be brighter, and they have a great potential to be more biocompatible, because we can even design particles that can really biodegrade, undergo biodegradability in the living system. And we spent quite a lot of time and efforts in study the biocompatibility of these nanoparticles. And I have to say that not all the AI molecules are highly biocompatible, but we are able to screen quite a number of decent molecules that are really very biocompatible, so that will allow us to actually move further into the biomedical research. So I would like to take this molecule as an example. This is the rate emissive AI molecules. And once it's formulated into nanoparticles about 30 nanometer, they are able to actually offer a unique opportunity, which I call the cross-platform fluorescence imaging. That means this molecule is compatible with many facilities on the market that allow us to do imaging. That includes in vivo imaging, cellular imaging, tissue imaging, multiphoton imaging, and even super resolution imaging. So the beauty of this molecule is that it allows us to actually obtain the information from both macroscopic to the microscopic, and with data that are able to validate with one the other. So that actually enhances our capability to produce imaging with a lot of information that are more precise than before, and much more with better possibility to actually cross-validate it than before. So because of time limit, I will just show you one experiment of how these molecules can be used to do something that is directly related to translational research. So we use AI molecule markers, AI nanoparticles to label the cells, and we also benchmark that with commercial dyes as well as quantum dots. And we show that we're able to label the cells in a way that's much more effective with higher brightness, so that we're able to track different generations much longer as compared to other commercial sources. So with this initial experiments, and subsequently what we did is to collaborate with clinicians and research staff from National Cancer Center in Singapore. So we take the tissues from the patients, and we use the nanoparticles to label the tissues, and then immediately we inject the tissue into the brain of the zebrafish. And we monitor at day 3 versus day 7 what is the survival of the cancer tissues that we actually, the tissue, not cancer tissues, that we inject into the brain. And it's very interesting that if these tissues are not cancerous, actually the capability for the cells to survive and graft into the brain vascular structure is very minimum. But if these tissues that we took from the patients is cancerous, then we find out something amazing, that we're really able to visualize that these cancer cells are able to survive and able to grow and really form something that are very well integrated with the vascular structure of the fish brain. So through this study, we're able to quantify basically the patient tissue, whether it's cancerous or non-cancerous, and this can all be done in 3 to 4 days. And compared to currently in the clinics, it takes about 2 weeks. So following that, we did another experiment, which is even more interesting. So we inject the tissue labeled with our particles into the fish brain, and we monitor how the cells actually migrate in the next 2 days. And we find out for the patient tissues which contains the cells that can undergo cancer metastasis, we will be able to observe the migration of the cells from the injection site to the body and even to the tail. And by monitoring this distance they travel within a certain period of time, we will be able to quantify and understand how vigorous or how severe is the cancer metastasis. So with this strategy, we're able to visualize the process of the cancer metastasis, and therefore we actually further did experiment to take in different drug and drug combination to use the same fish actually to figure out exactly how effective and what is the best optimized composition of drug that is most effective to patient. So we're still continuing in this direction and trying to develop a mature platform that's really be able to help the clinicians in make precise decisions for each of the patients. Now, for the same nanoparticle, just now the experiments I showed to you is really to track the cells and cell migration to visualize the cell. Now, if we incorporate the therapeutic functions into these molecules, immediately we can transform our nanoparticle from an imaging reagent again to an imaging and therapeutic nanoparticles. In this case, actually, we have done quite a lot of work with primarily focus on 2 types of nanoparticles. One is the nanoparticles with photosomol functions and together with the photoacoustic imaging. That's basically look into how the light absorption can effectively transform into the heat. And the second type is the fluorescence imaging coupled with the photodynamic therapy. So the example I want to share with you is the interesting one where we're trying to solve a key limitation of the light penetration. So in this case, we integrate an AI molecules, which is a photosynthesizer, together with a chemical, and this is a high energy chemical. And we fabricate the nanoparticle, and then the nanoparticle is emissive in the presence of the hydrogen peroxide, which is actually a cancer marker. And it also automatically producing the oxygen. And the mechanism is the following. So the CPPO actually reacts with the hydrogen peroxide to form a foam membrane that contains high energy, which actually releases the chemical energy to excite the photosynthesizer that gives out the light emission, as well as the oxygen production. So all these steps are instantaneous. As far as the chemicals meet the hydrogen peroxide, then everything will just happen like that. We conducted the animal experiments trying to use these nanoparticles to visualize and treat the tumors. And the first row actually referring to the chemiluminescence imaging, and the second row referring to the fluorescence imaging for the same mice with the same dose of the nanoparticles. So it's very clear that the chemiluminescence offers much higher selectivity in terms of the tumor imaging. And because these nanoparticles really functions like a very intelligent rockets, they will go attack this tumor site to light up the fluorescence, and at the same time, they will be automatically releasing the oxygen to start to kill the tumor by themselves. This is indeed very effective. If we monitor this whole process for about 2 weeks, and we indeed find out with these nanoparticles, they're able to find the tumor, they're able to kill the tumor, which means the inhibit of the tumor grows very effectively in 2 weeks' time, so that we're able to elongate the lifespan of the tumor-bearing mice with the treatment. And very recently, we went one step further, trying to solve the problem of how to treat cancer metastasis. So we built an animal model where we have the primary model, we have the primary tumor, we also have the secondary tumor, and we're trying to cure the mice actually by working on the primary tumor. So that's why we developed a dual model reactive oxygen species driven tumor immunotherapy process. So in this case, we treat the primary tumor with high power near-infrared irradiation, which actually induce the immunogenic cell death and release the antigens. And our particles are designed in such a way that it will be able to capture this antigen and then carry them to the nearby lymph nodes. And there, we will use the subsequent lower energy near-infrared irradiation so that it will activate the dendritic cell and basically boost up the overall immunoresponse of the animal, so that we're able to not only cure the local tumor recurrence, but also prohibit the distant tumor growth. So this whole article took us quite a long time to finish because of extensive, large amount of biological experiments. So I will not elaborate more. So for those who are interested in biology, this is really an interesting story to show how materials can make an impact in solving complex biological problem. So we'll come to the last piece of small story. So as we work on the chemiluminescence, we ask ourselves whether we have other modes of imaging so that we'll be able to provide a better contrast and really eliminate the background signal of the biological species itself. Then we start to focus on the phosphorescence. Because of the long lifetime of the phosphorescent molecule, we actually can have a time-gated imaging. So what I show here is that if we look at cell imaging with the phosphorescence versus non-phosphorescence, there's a huge difference in terms of the background and the contrast. And if we look at in vivo imaging, it becomes even more obvious that if you look at the phosphorescence imaging, it's very clear signal with zero background. And if you look at phosphorus imaging, it's like everywhere bright because of the autofluorescence of the living system. So this drives us to look into the photophysics of the molecules. And basically, we look into how we can design organic molecules with room temperature phosphorescence, because we wanted these molecules to be applicable at room temperature in the presence of water and in the presence of oxygen, which is kind of against nature because triplet state is very sensitive to oxygen and to moisture, etc. But this is something that's really interesting and that really can happen. But before we get there, we find something more interesting and something that really come to us as a big surprise, which is the carbazole as a starting material. So we designed a series of molecules which contain carbazole in order to realize room temperature phosphorescence. And we find out if we order the carbazole from commercial sources, it actually shows beautiful fluorescence and phosphorescence. But if we purify further, and we find something funny. So this phosphorescence is not stable. It actually varies depending on how we purify it. And then we decided, how about we resynthesize the carbazole from all the smaller pieces of molecules, and we find something quite shocking, that the phosphorescence phenomenon almost disappears. And we did detailed analysis using HPLC and also grew the crystals, and took us quite a few years to identify that commercial carbazole actually is accompanied by a very, very small amount of isomer. They are exactly the same molecular weight. They are very similar optical properties, and they're very difficult to separate from one the other. And with an extremely small amount of less than 0.5%. But this molecule exists in almost all the commercial carbazole molecules we tested. And it's showing a tremendous impact on the optical properties of the molecules. And I would like to elaborate slightly more. So now, if we synthesize this molecule, carbazole, actually from scratch, it showed the emission maximum at about 360 nanometer. And if we look at all the carbon we get from all different commercial sources, actually they're exclusively showing the emission at about 420. So that's something quite interesting and quite shocking in a way that they're very different. Now, if we use the lab carbon plus this isomer, we build a standardized curve to look at HPLC so that we are able to quantify the amount of impurities from each of the commercial sources. And we find out that for all the major commercial sources we tested, it contains the impurity in the range of 0.1% to 0.5%. Now, if I use commercial carbon to just look at the phosphorescence, the first row is what we bought from commercial sources. And then we conduct a few steps of purification, such as crystallization, column, and then we find out the crystals actually become a lot brighter because during the process, carbon and isomers still stick together, but other impurities have been removed during the process. But if we look at the molecules that are synthesized from lab, we barely been able to really visualize this with our naked eye or camera in terms of the ultra-long phosphorescence at room temperature. Now, interesting enough, if we mix these two molecules together at different ratio, and both carbon and his isomer really do not show any ultra-long phosphorescence or ultra-long emission, but when we mix them with a very small amount from 0.5% to 1%, and sometimes even 0.1%, we're able to significantly boost this ultra-long phosphorescence at room temperature. And we tried many other compounds, and we observed similar phenomenon that if we synthesize them from commercial sources, they show all this beautiful phosphorescence. But if we actually resynthesize everything from scratch, we lost this function. And we're able to bring back this function if we use this impurity derivative to actually dope the carbon derivatives. So this is another example that we show both molecules actually do not have ultra-long phosphorescence at room temperature, but as we mix them, we are able to almost something like we're almost able to transform the fluorescence molecules into phosphorescent molecules as far as we just mix them at different percentage of the dopants that we discovered. And of equal importance is that it is generally believed that because of the phosphorescence to occur at room temperature is very difficult, so that I think the general belief is that this molecule has to exist in the crystalline state with really highly restricted molecular motion so that we will be able to observe any room temperature phosphorescence. And what we find out here is that if we use this host molecules and mix together with the isomers, even when they're in an amorphous state, they are still able to show ultra-long phosphorescence at room temperature. So that really kind of changed our mindset that some of our concepts are too rigid or too demanding in order to obtain certain molecular behavior in the literature. So we further conducted a series of experiments trying to understand what is the mechanism, and we proposed the trap mechanism via 3 stages. In the first stage is the charge transfer stage, where we use transient absorption to actually, we're very lucky to be able to capture the carbazole ions, which is the carbazole negative, and so that the isomer will be able to form a positive ions. And in the second step, it will undergo a charge separation. So that basically is a process which allows this emission to come out gradually over a long period of time in seconds or tens of seconds. In the third step is the charge recombination. And in our system, when the 2 positive and negative charges come into combine together, actually it allows both phosphorescence and the delayed phosphorescence to happen, so that in this system basically is a dual emission of both phosphorescence together with the fluorescence. So with this, I would like to summarize the AIE molecules. So today I had this great opportunity to share with you some of the examples of how AI molecules can be designed and can be utilized to address some of the biomedical issues. And actually, AI molecules and the AI community has grown tremendously over the past 20 years. And I work very closely with Benzong Tan, who is a professor from Hong Kong University of Science and Technology, and he's also the champion in this research field. So AI molecules has developed into a really fascinating subset of materials, which have very unique properties and applications in light emitting devices, in visualizing chemical and biological processes, in sensor applications, as well as in the biomedical research field. So with this, I would like to summarize that today I talk about the molecules with the aggregation-induced emission. And over the past few years, we primarily focus on 4 important concepts: AI light-up probes, AI DOTs, AI photosensitizers, and AI biopsical probes. And we're managing to gradually, step by step, to push each of these discoveries into commercial products. And in terms of applications, we primarily focus on detection, imaging, and imaging-guided surgery and therapy. So if you're interested in knowing more, you can refer to some of these review articles. I'm very grateful to my collaborators, my research team, and also the funding agency. So before I take your questions, I would like to spend a minute or two to really talk a little bit more about the exciting journals that I'm looking after. So ACS Materials Journal, Materials Letters actually is a sister journal to the chemistry materials. So we take care of communications, and chemistry materials take care of the 4 articles. And we focus on exciting transformative material research. We have been publishing, I think, quite a lot of issues because we're already 1 year old. We really look for high quality, urgent contributions with exciting materials that are able to help us to transform not only materials research, but how these materials are able to help us to addressing key challenging problems associated with environment, associated with healthcare, associated with energy, and associated with many, many exciting things that we really care about on a daily basis. We're very special that we offer immediate pagination, means as far as we accept your article, you will get your page number immediately. So I really hope that you will take a look at our profile and look at the wonderful authors who publish with us, and then submit one of your best research to us today. So if you are interested in understanding more of this journal, we do have a very vigorous and dedicated service actually to promote each of the journals in English, in Chinese, and in other language through the Twitters, WeChat, and other media. Thank you very much for your attention.
Okay, I really appreciate the great research and the excellent talk, and also the nice advertisement. And
Thank you.
We have a few minutes for the questions. So the first question was given by An Jae-hwan. Thank you for your talk. Could you reiterate the mechanism of triggering the aggregation behavior upon the addition of water?
Okay, so basically, a standardized experiment that everyone can try is that you have a molecule in your hand, you dissolve this molecule in tetrahydrofuran. Actually, can be other solvent that is really miscible with water, and you make sure that at the molecular species, when the molecule is dissolved in a good solvent, is in a molecular species, then you will be able to understand the optical properties of this molecule as an individual. And then you gradually adding water into this tetrahydrofuran solution. And upon a certain volume fraction of the water, you will be able to observe a change in the fluorescence intensity. And if you monitor what's the change in the solvent or suspension using, for example, light scattering or AFM or TM, you should be able to see that as the fluorescence changes, the size of the nanoparticles or the aggregate is formed in the solution, so that you can correlate your optical behavior with the change in the molecular aggregate in the solution. And then you will be able to draw a conclusion, yes, this is aggregation-induced emission, or no, this is not aggregation-induced emission.
Okay, thank you. And next question. So thanks so much for your great talk. I'm not familiar with your topic, but is there any issues on nanoparticle toxicity for cancer or other body healthcare application? I guess you already showed some data.
Yeah. We care a lot about the toxicity of the particles because we rely on these nanoparticles to imaging and doing therapy. So we need to make sure that first, these particles need to have very good biocompatibility, especially when you use them as a contrast agent to visualize anything wrong with the body. We cannot stand in something toxic to visualize something wrong with the body. Second, even as the therapeutic reagents, we also need to make sure after the treatment, this nanoparticle either should be cleared from the body very quickly, or they will undergo biodegradability within the living system to become smaller fragments that they can come out of the body very quickly, or we're able to make the nanoparticles so small that they will come out, let's say, from the kidney. That will be a pathway that's more effective as it comes from the liver, which takes much longer time to clear. So biocompatibility actually is one of the most important and top priority parameters that we evaluate each of our nanoparticles before we use it for any in vivo applications.
Okay, thank you very much for your great talk again, and I hope you to meet here at KAIST again. Okay, thank you very much.
Yeah, thanks.